Alright guys, I'm going to give you a little review of Chapter 14, Lesson 3. This is called the First Life Forms. I know that I'm going to move probably a little quicker than you can write throughout this whole PowerPoint. Don't worry, the whole thing is posted on my website so you can copy everything down into your notebook, so don't worry about writing like super fast. Nevertheless, we talked in a previous chapter, which you had your last test on, about transcription and translation. We had our DNA and it was going to RNA for transcription, and then it was going to a protein for translation. What people started to wonder was that, okay, we got DNA, it has all of our information that we need to make a protein, but we have this middleman, this RNA in the middle. So scientists came together and they kind of went over, all right, if we have the instructions in the DNA, why do we need this RNA? They found that maybe this RNA was going to have a little bit of a deeper meaning. And what they found was, if you remember, whenever we have the mRNA bonding to the tRNA, they found that the hydrogen bonds in between that and also in the tRNA between all the base pairs we're actually going to change the shape of the RNA, and they thought, well, maybe this plays a role in this whole big picture. In order to give you guys a real good visual of this stuff with all the hydrogen bonds, I figured we would relate it to proteins because I think that's the easiest thing for you to visualize. So let's say we got a nice big string of amino acids there. Now, if you remember, all of our amino acids are held together by peptide bonds. These are pretty strong bonds. But that's not the only bonds that we see in polypeptide chains, in proteins. There's also other ones, and if you see here between our two CYS amino acids, that's a hydrogen bond there with the two S's on it. It's a hydrogen bond being made, and that's caused by, if you remember the structure of a protein, that R group on the top, that variable group. There's polar-polar and polar-nonpolar and nonpolar-nonpolar interactions. I know that was a mouthful between our amino acids, but with those polar-polar amino acids, they are going to cause these hydrogen bonds to form between them because polar and polar, they attract one another. And this is what causes the whole protein to form in the shape that it does. It's almost like whenever we talk about enzymes, which are a type of protein, they fold in a certain way to make that active site so that our substrate can bond to it. But we'll go over that a little bit more later. So as we were talking about the enzymes, we know that enzymes are going to catalyze chemical reactions. If you remember, this old picture here with the active site we just talked about on the last slide. But they were thinking maybe these RNA molecules do the same thing and they're able to also catalyze chemical reactions. So what happened was in the 1980s, a guy named Thomas Sech, he came and did a few experiments, and he had this theory inside of his head. He said, well, maybe some of these RNAs can act like catalysts just like enzymes do. What he called his theory was a ribozyme, and what a ribozyme was is pretty much RNA that acted like an enzyme. It had the same function of an enzyme. Just to kind of recall, guys, from 2 chapters ago now, if you remember, we had the RNA Primer. I know this is like a tangent away from Sech, what we just went over, but it'll make sense in the end. Remember the RNA Primer? That was needed in order for DNA replication to occur because the DNA polymerase, remember that added all the nucleotides to our template strand to make our new strand of DNA. In other words, it paired complementary base pairs by adding those nucleotides. It needed that RNA primer so that the DNA polymerase had something to bond to so that the DNA polymerase could go and put all those nucleotides in. So what they thought was that since our cells in order to reproduce, needed a duplicate of the DNA, and that duplicate of the DNA needed the RNA primer, they knew, all right, without RNA, we're not going to have any more cells. So they thought, hey, makes sense. RNA must have been there before DNA was. Another hypothesis is that early forms of RNA had to compete with other RNA In other words, it was like survival of the fittest. There was, think of a big swimming pool of nucleotides, and we had all of these RNAs, and they were competing against one another so that, one could have a full strain of nucleotides and complete the whole RNA strand. Those ones were considered the fittest of all of the RNA, and they were able to survive. Ones that did not get enough nucleotides to fill the whole strand obviously weren't able to survive. So this kind of gave a little bit of differentiation between our RNA. So now, off that tangent, back to Sech, he later found that the ribozymes could catalyze their own replication. So this further supported the fact that, all right, this RNA was enzyme-like, and it definitely was here before DNA. So pretty much what was happening here, guys, was that the ribozymes were able to replicate themselves and make more ribozymes. Pretty efficient way of doing things. So this brings us to our first cells. Scientists had three kind of inklings, inferences back in the day. They thought that when Earth was created, there was little or very no amount of oxygen that existed. They thought that with this, since there was no oxygen, there's probably not going to be many organisms that go through cellular respiration like we do because we need oxygen. This also brought them to the assumption that probably the oldest living things were prokaryotes. They've just studied a bunch of fossils and they found that out. The third one's the most interesting. The first cells probably developed in an environment where it was probably a very extreme environment. It was probably very hot, maybe slightly volcanic, and they thought that these first organisms actually were anaerobic, obviously because there was no oxygen. They can't be aerobic, they can't go through aerobic respiration, and they were also heterotrophic, which means they could not make their own food. This kind of stunk for them because they were competing for their food, but it was really good for the autotrophs because the autotrophs, they did not have to compete for food because autotrophs make their own food. So it gave the autotrophs a real advantage of surviving because they were kind of laughing at all the heterotrophs, not literally, but they were laughing at all them being like, oh, you guys have to compete for your own food, I'll just make my own over here. The next thing with the first cells that your book goes over is the chemosynthesis. What they found was that there was a group of organisms that they found in different fossils, and they thought that they were really closely related to a group that we know now as archaea. This is unicellular, and they live in very harsh environments, and they're also autotrophs, which means they make their own food. Relating to the chemosynthesis, what they thought was these archaea obtain their energy by this process. So what they were going to do is take carbon dioxide, which is not oxygen, complete opposite of it, right? And they're going to use the carbon from the carbon dioxide, and that's going to serve as an energy source for them to make up organic molecules, whether they were proteins, lipids, carbohydrates, or nucleic acids for the organic molecules. So they didn't think that photosynthesis existed until about 3 billion years ago. So the first kind of billion of years, there was no animals that went through photosynthesis. I shouldn't say animals, plants that went through photosynthesis. But later on, our photosynthesis plants, or even algae, was able to take all of these carbon dioxide molecules. They were able to bring them in, and then they were able to convert them to oxygen, and then they started giving off oxygen. That's kind of what plants do today. It's exactly what plants do. That's why we are able to live how we live. They looked at some of the fossils, and they found that some of the cyanobacteria was probably some of the oldest photosynthetic organisms. They were unicellular, and they were able to give off oxygen. However, they didn't think oxygen came around as the way it did today until many years later. It was very slow to come about, all right, but it did start to lead to the formation of aerobic respiration, all right. It did take a whole lot of time, though, So we talked about it was 3 billion years ago for those, but the atmosphere isn't it doesn't have the amount of oxygen that we have today until about 1 billion years ago. So there was about a 2 billion time period where they did not have the amount of oxygen that we have today. Something cool about the oxygen is in our atmosphere, and we've got a bunch of different layers, closest to us, closest to the Earth, the surface of the Earth, we have the area where oxygen is how we know it today, the oxygen we breathe. This is 2 oxygen atoms bonded together. As it goes higher and higher and higher, closer to the outer atmosphere, what happens to that oxygen? It becomes kind of radioactive, I guess we can say, just for lack of a better term, and it bonds to other oxygen, creating 3 oxygens, which is not the oxygen we breathe. It's like heavy oxygen. We call that molecule ozone. All right, so we can't breathe that in. It's up high, it's way, way above us, so why do we have it? That ozone, something kind of cool about that, is it actually helps us to not get ultraviolet radiation from the sun. If we were to get ultraviolet radiation from the sun, our DNA would be damaged, and yeah, nothing on the whole Earth would survive. There's the layers you guys could see. The troposphere is where our oxygen is kind of how it is today, and then the ozone layer above that is where we get that O3. All right, last few slides, guys. Lynn Margulis proposed that eukaryotes evolve by early eukaryotes engulfing prokaryotes. This is called endosymbiosis. So what we had was a prokaryotic cell. It developed a nucleus, and then it started to take in other prokaryotes. These other prokaryotes were going to be the mitochondria and the chloroplast. So with the mitochondria and the chloroplast, if you remember what we went over a long, long time ago, those mitochondria and the chloroplasts, they have a double membrane, and they also have, along with the double membrane, their own DNA. With having their own DNA, the cool thing is they probably were prokaryotic back in the day. So whenever the small prokaryotes, the mitochondria, the chloroplast, came into the eukaryotic cells, they kind of had a symbiotic relationship with them. By this is mean like each one of them is getting some kind of mutual benefit. So what happened was, whenever they came in, the prokaryotic cell was able to have a very nice stable environment from the eukaryotic cell. However, going on the other side to make sure the eukaryote was benefiting as well, if you remember, the mitochondria makes energy for us, and the chloroplast for a plant cell, obviously we don't have chloroplast, but for a plant cell is going to bring in that sunlight. So that's kind of how we came from the prokaryotic cells. It was kind of nice because we have a mutual relationship with them. So anyway, guys, hopefully this helped with section 14.3. You guys can copy your notes down, and I'll see you later.